Hello and welcome to today's webinar, which will be presented by Alexandra Graff-Meyer. The title of her talk is Serial Block Face Scanning Electron Microscopy for Large Volume Brain Tissue Imaging. Alexandra studied biology in Strasbourg, France, before completing her PhD in 2004 in nanocarriers for drug delivery in Basel, Switzerland. Her first postdoc was at the Maurice Müller Institute under the supervision of Professors Uli Ebbe and Peter Burkhardt, where she characterized peptide-like particles for biomedical applications such as drug delivery systems and vaccines. In 2008, she started her first project with electron microscopes at the Center for Cellular Imaging and Nanoanalytics, also in Basel, Switzerland, first under the supervision of Professor Andreas Engel and then Professor Henning Stahlberg in cryo-electron microscopy. She was involved in single particle projects dealing with various proteins and established a collaboration with Roche Pharmaceutical Company, providing electron microscopy services, developing her service-oriented skill. In 2014, she joined Christelle Genoux at the Facility for Advanced Imaging and Microscopy and became a permanent member in the 3D electron microscopy group. Since then, she has provided cellular electron microscopy services for FMI and Novartis researchers. Alexandra will be available to answer some questions at the end of her presentation. To ask a question, please enter it into the chat panel of the GoToWebinar user interface. In case of any connectivity issues, please also use the chat window. We will attempt to address these immediately. But for questions about the topics discussed today, we will answer those at the end of the presentation. And with that, I will hand you over to Alexandra.
So welcome all to my presentation. And let me first thank Gatan team for organizing the webinar and for giving me the opportunity to share our serial block face SEM workflow for large volume brain tissue imaging. The scientists at FMI are motivated to understand the basic molecular mechanism of health and disease in the field of neurobiology, quantitative biology, also in epigenetic. In addition, the FMI is hosting 9 technical platforms that support scientists with their research questions, providing them expertise, high-end instrumentation, and technical support. Our imaging platform provides technical expertise in electron microscopy, light microscopy, eye contact screening, also in image processing, and we have the chance to have an IT member who provide us services in logistics for our instruments and provide strategies and solutions for handling big data, for example. Our electron microscopy platform offers services and training going from sample preparation to image analysis. We propose various EM technical approaches going from 2D transmission electron microscopy for ultrastructure or immunolabeling investigation. In addition, we provide 3D electron microscopy services with serial block face scanning electron microscopy for Connectomics. The technical expertise in 3D electron microscopy was introduced and implemented by Christelle Genoux together with Rainer Friedrich and his group. Over the years, the technique has been a lot challenged by his scientific question in Connectomics. For example, the group of Rainer Friedrich want to understand how the brain processes and computes information. To address this question, they need to reconstruct brain neuronal circuit by combining analysis of connectivity called Connectomics with studies of circuit functions and behavior. For that, they use the olfactory bulb of zebrafish larvae. They use this larvae as a model system. To achieve this, they correlate neuronal activity pattern of the zebrafish olfactory bulb using multiphoton calcium imaging together with the reconstruction of neuronal circuit, neuronal connection, using serial block face scanning electron microscopy imaging. To record neuronal activities, they perform 2-photon calcium imaging, and for that, they immobilize larvae, zebrafish larvae, into agarose, and they submit the larvae to different odors. Once the larvae is stimulated, it produces an activity pattern, and this is done multiple times for many hours. Different type of odor are submitted during this experiment, and it gives us a rise into a certain activity pattern. Once this part is imaged, exactly the same larvae is used to perform electron microscopy imaging. And for that, the larvae is then dissected, and they do a little craniotomy to facilitate the region penetration for electron microscopy preparation. Then the sample is fixed and prepared for electron microscopy imaging. Why do we need a serial block phase electron microscopy for reconstructing connectivity? In fact, there are different types or different modalities to perform 3D electron microscopy imaging. We have electron tomography, serial sectioning, SEM, and array tomography. But the serial block phase scanning electron microscopy offers unique pictures like large field of view, and in the meantime, we gather very high resolution images, and in addition, the slicing in Z is lower than 30 nanometer. At FMI, we are lucky to have two systems hosting a 3-view serial block phase microtome. One is the Zeiss Merlin and is only working in eye vacuum, whereas the Gemini is much more flexible and has different options, and we can work in high vacuum and also in variable pressure. In addition, we customize this microscope with 5 megahertz amplifier to be able to scan 4 times faster. And we are also having the focal charge compensation system to prevent charging of the sample. Now, let me go quickly how the microtome is mounted in a SEM. As you see, the microtome is mounted on the door of the SEM, and you see on the middle part, so you see the sample in the holder. This is the microtome with the knife, and the block sample is positioned directly under the SEM column. After imaging the block face, the sample is moved up in with very small step down to 25 nanometer. The ultramicrotome knife cuts the top of the sample and retracts, exposing a new block face. Then the process repeats. And in this way, you will cut and image thousands of blocks, faces, and reconstruct the data to a complete 3D volume. And here you can see a scheme of the acquisition. So now the beam is scanning the surface, the knife is cutting, scanning, cutting, and so on. Here you see in the middle the block face. This is this black area surrounded with silver epoxy. I will come later with this detail. Before we go on with example of imaging with scanning electron microscopy, let me give you a little brief basics about how a SEM is working. So you need the electron source, which is on the top of the SEM. This beam is then produced and need to be focused until it hits the sample, which is positioned here. The beam is then rasting the surface of the sample, and the electrons that are scattered from the sample are converted in gray value, pixel per pixel, to produce the image. Let me talk about the electrons we are collecting for producing the image. We use in serial sectioning, in serial block phase, backscattered electrons. Those electrons are very high energy electrons, and they are almost not deviated from the primary beam. Here you have a scheme of how it looks like. So you have the beam and the electrons shoot the sample. They are backscattered and detected by the backscattered electron detector. But imaging with backscattered electron can be very challenging. And because the backscattered electrons are not much deviated from the original beam, the backscattered detector must be mounted above the sample. Also, the backscattered electrons are high energy, and for biological sample, it's required to image at low kV. Those are views of the Gatan backscattered electron detector. Here it's actually the detector that is mounted directly on the objective lens from the microscope. Here you have the view of the stub, the knife. So there are little space between the knife and the sample and the detector. And this is reality. That's pictures I took with my phone. And you see, actually, there are very, very little space between and the detector and the knife that actually is retracted. So little space between the sample and this backscattered electron detector. So always be very careful when you do the approach and when you mount the sample. And one of, I would say, the problem is we are imaging biological samples who are light elements, so low atomic number, and those samples are very poorly conductive. Backscattered electrons are very well interacting with heavy, very high atomic atomic number, and that's why we have to prepare the samples to make them conductive. Now let me present you the sample preparation part, which is a very crucial step for serial block phase imaging. I would say it's almost the most critical part, not critical, but the most important part for being able to image the sample. To prepare the tissue, in the past, we were using the Rank and All protocol from 2010. This protocol has really worked very well for mouse brain, and actually also for the olfactory bulb from the zebrafish. But you will see later that we had to adapt it or create a new protocol for different part of the zebrafish brain, but also for different stage of the zebrafish. So the protocol from the Rank and All is an accumulation of heavy metal on the sample. So it's different steps of osmium. In addition, you have this thiocarbohydrazide that bridge the two osmium steps to make more and more layers of heavy metal in the sample. In addition, we have a step of uranyl acetate, and finally, a final layer of lead aspartate make the sample even more conductive. After dehydration, the sample is placed into resin, and this is how the fish brain looks like after being prepared with the sample, with this protocol. In addition, we are embedding the sample in a silver resin or epoxy, and this also prevents additional charging that may not be cancelled by the sample preparation. After that, it's mounted on a pin, and we can start the acquisition. So what you see here, it's the overview of the olfactory bulb from the zebrafish larvae, surrounded by the silver conductive epoxy. And that's a zoom, and you see nicely the glial cells from the olfactory bulb, and they are not charging at all. Now, I would like to show you how the staining can influence the different parameters of the acquisition. And once you see, when you have no stain, it's almost impossible to image at very high resolution. You have to compromise the cutting, but also the resolution X and Y, and it required very long dual time, and it's almost impossible. You need to image at high vacuum. It's impossible to image at high vacuum. You need low vacuum to be able to image some sample. And by increasing the amount of heavy metal in the sample, you see that the conditions for imaging are improving more and more. And that's now our standard acquisition parameters. So we can even acquire at lower kV, and we can reach in X and Y even lower, 10 nanometer pixel, and cut in Z at 25 nanometer. Actually, I forgot to, it's a mistake. Actually, it's microsecond, not millisecond. That's way too much. Sorry. So the art of sample preparation. We don't have the magic solution or the magic protocol. I mean, you have to explore the literature and to understand the different steps of the sample preparation before you go with your own sample. You have also to understand the inevitable artifacts you are generating. And you may develop your own flavor of protocol to best preserve the sample and for imaging your type of samples. I think that's the take-home message regarding the sample prep. Once the sample is ready for acquisition, we want to have the sample acquired in a very automated way. And that was our motivation to develop software to control the acquisition of the sample in a semi-automated way. And Benjamin Titzer joined the group of Rainer Friedrich and developed SBEMimage, which is a versatile acquisition software for, say, a block-phase electron microscopy. Typically, in the Gatan configuration, the digital micrograph, this is the software that allow the acquisition of images, control the microtome, and control also smart SEM. What we wanted is to improve the monitoring and the error handling. We wanted also to have additional options with the program. And for example, debris detection and having also an option for removing the debris was one of the idea of Benjamin to implement in the SBEMimage. We wanted to have a higher effective acquisition rate, and we wanted to have software highly flexible in operation and also very extensible, meaning it provides a platform to that the users or the SBEM users could implement new option into that software. So this is how it looks like. So actually, so the SBEMimage control in parallel, smart SEM and digital micrograph, and is communicating between both hardwares. So if you are interested by this open source software, this SBEMimage was released to the community, and it's an open source project on GitHub. So this is the address. You will find additional information regarding this software in his paper, Titze and al. From 2018. And I forgot to mention that this software was written in Python. Quickly, let me just show you the interface of this of the SBEMimage. So it's composed of a viewport where you see an overview of the sample. Here we call this a stub overview. On the right side, you have the main control. So here you have different windows. I don't want to go today into all the details of this. I think it's very well described in the paper from Benjamin. But I mean, I will show you just two options that are very useful for semi-automation of the acquisition and why we implemented this. So let me quickly finish with the viewport. What you see, it's the stub overview, and in the background, you have also the silver, so you recognize the silver epoxy. The grid are produced via the main controller, and here you can select which tile or which part of the brain you would like to image. By selecting the tiles, you tell the microscope to image this area and so on. So you can produce different grid and image in parallel different area from the brain. On the fly, you can also, once you are done with this area of the brain, you can unselect easily the tiles. So we have a very flexible system to acquire or not acquire the tiles. We call this adaptive tile selection tool. We have also the option of monitoring the data, the quality of the data. And for this, we have the slice viewer, which is very convenient if you want to check if the sample is properly cut over the different sectioning steps. And it allows you also to look at the full resolution of your image, which is important, and also to assess the quality of the tissue and also, yeah, the cutting quality. You have also a live quantification, and you can also observe the histogram of the image, and you have image statistic and reslices. In addition, when you are doing very large and long acquisition, you would like to also maybe do that remotely. You don't want to sit all day long in front of the microscope. And for that, SBEMimage is very useful because the idea was to remotely control the focus and the stimulation from your computer. And for that, this was implemented also in SBEM. now we are ready for acquisition, and I would like to show you an example of a stack we acquire now a few some times ago. This is Olfactory bulb. The acquisition is 1.5 kV with dual time beam current of 300 picoamp. The staining is with the Derenk protocol, and this has lasted 4 weeks for this acquisition. That's not our longest acquisition. Actually, the longest we did in the past was 14 weeks, non-stop acquisition. Once you are done with the 2D, the 2-photon calcium imaging, and you are done with your stack acquisition with serial block phase, you have to correlate the 2 stacks or the 2 modalities of imaging to be able to reconstruct the full circuit. And for that, the group of Rainer Friedrich used a landmark-based image registration procedure. So you see here, in the back, you have the image of or overview of one slice, and this is acquisition from the 2 stack from the 2 photon imaging modality. So after alignment, after acquisition, you would like to do alignment, and actually you don't need so much alignment because the images are very nicely overlapping to each other. So you proceed you need to proceed with a translational alignment procedure. And then you proceed with the group of Rainer choose a strategy of using custom MATLAB program to pre-process and do registration and stitch images. And the same for image registration. This is explained, all this alignment workflow and reconstruction and analysis in the paper from Wanne and al. Published in 2016. I don't want to spend too much time on that because I'm not a big expert in this, but I think it's worth to mention it. Once you have this aligned properly, then you can proceed to the alignment. Adrian Wanne, a student from Rainer Friedrich, has now his own startup and provide service for segmentation of neurons, and you can find more information on that website. This software that Adrian developed, or he didn't develop those software, but he used those software for doing 3D image annotation and neuron reconstruction. Once you have done all the steps, a very long procedure, you end up with a beautiful diagram of neurons and synaptic connections. Yes, I forgot to quickly show you how it looks like a synapses, but I think maybe you know all how it looks like. That's just a scheme how synapse contact cells make contacts and how they talk to each other. So finally, this is the 3D reconstruction of connectivity in the Zebrafish olfactory bulb. And I hope you appreciate this crazy work of reconstruction. And by zooming in, you see all the details from the reconstruction and the wiring, and the colors. Color code is for the different type of neurons. Okay. Now, this all workflow have been done by Adrian Wanne. But Rainer Friedrich was also very interested on developing this similar workflow for the adult zebrafish, but also to image other part of the brain. So for that, we proceed like Adrian Vanner did. We used the Ellisman protocol or the rank protocol, and we exactly use the same workflow. But what we realized is that this the rank protocol was absolutely not adapted for this new type of tissue. It's not a different tissue. It's just that the stage of the fish larvae, zebrafish, requires maybe a different preparation. So you see here that's the part of the cortex or telencephalon, and you see this gradient of stain, very nicely stained around the surface of the tissue. But as soon you go deeper in Z, you see very light piece of a part of the tissue, and here you see even charging at this magnification. For this reason, so we observe staining gradients, also charging issues, and when we were zooming into the different part of the tissue, we also saw that the ultracellular space was not preserved. So our objective for this brain, tissue adult brain, was to have a protocol who gave us a very homogeneous staining over the full sample. We needed to improve the contrast to image at a very low KV. We need also a protocol that gives us sufficient conductivity and give us excellent preservation of relevant structures. And also we needed reproducibility. And we achieved that by being inspired by the protocol from Mikula and Denk, who developed the protocol called BROPA. For this BROPA protocol, called brain-wide reduced osmium staining with pyrogalol mediated amplification, was reduced for our application to 6 days, and we introduced, in addition, a lead aspartate step. You see that in this protocol, BROPA protocol, there is no uranyl acetate, and I think that's very interesting for people who knows radioactivity. And using this FBROPA protocol developed by Genoux and all, we managed to have we managed to have a tissue. This part of the tissue is homogeneously stained on the surface and also in the middle of the sample. This is an illustration of the proper staining we get with this BROPA protocol. And I hope you can appreciate the synaptic connections. And you see here the button synaptic, the connection between two synapses. And yeah. So we end up with a protocol, the FBROPA, having very good tissue quality, but also a good conductivity. And you see at the very near surface, this staining is very nice. And after you cut 300 microns deeper into the sample, you appreciate that also the stain is very nicely homogeneous. Here it's again a video of stack acquisition of this telencephalon part of the brain in the adult zebrafish, you may notice that the nucleus are charging. And this brings me to the next step. Sometimes you may have the best protocol, you have a wonderful contrast, but still you have some charging issue. And now it brings me to the part where you could compensate for charging. You see here two images of the zebrafish brain, and when you zoom in, yeah, you see that the image is quite distorted and very blurry. Either you go back to sample prep and you develop a new protocol, but in our case, it was not a no-go. Actually, we developed this FPROPA protocol on naive fish, fish that were not calcium imaged. Why? Because we don't want it to use very precious samples prepared with calcium images to develop this protocol. And we actually realized that sometimes the samples who were submitted to calcium images were a bit more prone to having charging. So sometimes charging issue is not fully solved, although you have a good sample preparation, and sometimes the tissue is not optimal due to the calcium imaging. The consequences then is that you may have uneven scene sectioning. You also reduce the resolution of your acquisition. You increase the signal-to-noise ratio, and the speed of acquisition becomes slower. So one solution is to, after retrimming the sample, it's to re-embed. So I already mentioned this embedding that solved in the past many issues with the olfactory bulb. But actually, since we have this new tool or new option, the focal gas assisted charge compensation, we were able to image even charging samples. And that's why we have not made big effort to redevelop a new protocol for the new samples. And now for very light issues with conductivity, we can use now the focal CC. Focal CC, it's actually a little flow of gas that is displayed on the surface of the sample or just above the sample surface to neutralize charging of the sample. You can find details of this focal CC in the paper from Tom DeRank, and this focal CC is commercialized by Zeiss. This is to show you the impact of charging issue. And in the past, we were used to compensate the charging by acquiring at variable pressure. But for our application for reconstruction of neuronal circuit, it was a no-go. We required the Z resolution and in lower than 25, and with variable pressure, that was impossible. So, and you see also the signal-to-noise ratio is very high. And once that's in high vacuum, and once you use the focal CC, you end up with this resolution. Always better to image at high vacuum, but if you have little charging issue, you have the possibility to use the focal CC. Quickly, let me go. So sometimes the charging can be very dramatic. And you see here, no way to image when you have samples with a lot of resin around. And once you activate the focal CC, you see the charging is completely cancelled. And that's a zoom of this area. And the same with this sample, you see that nucleus surrounded by a lot of resin, and we see that we really compensate completely for the charging. So that's a summary for how you can compensate the charging. You can compensate on the sample level by making the sample more conductive during sample. So you have to either develop new sample prep, and this is by far the best approach. And this is what we did also for by developing the fast BROPA. You have the possibility to use conductive embedding. Also, there is a way that we are not using in our platform. On our platform, it's to sputter a conductive layer on the scan surface. And of course, you can always try to optimize the beam energy to achieve charge balance or get closer to charge balance. You can also act on the microscope level by either doing the acquisition at low vacuum. That works well, but for the signal to noise ratio, you saw this is a no-go. And finally, since 2018, you have the possibility to get this focal charge compensation system from the rank, and that has solved a lot of issues for our samples. And quickly, let me talk about how we are also performing higher speed acquisition. A year ago, we got a 5 megahertz amplifier that we installed on the side of the Zeiss microscope. So it looks like this. This pre-amplifier allows us now to perform scanning speed of 0.2 microseconds. So we improve by 4 the acquisition time of our images. And I think for very long acquisition, that's very precious time. And no compromise on the quality and resolution, we are able to scan at the same dose and the same voltage. So no compromise. And with this slide, I would like to thank you all for your attention. And I would like to thank you in particular, the group of Rainer Friedrich, who really helped us to develop the whole workflow together with Christelle. Christelle unfortunately left us for a new challenge in Lausanne. She has now her own group in Lausanne. And I would like also to thank all the FME imaging facility for their support and for really this very nice team spirit.
Thank you, Alexandra. That was a really interesting presentation. We do have time for a few questions. To remind the audience, if you'd like to ask a question and haven't yet, please enter your question into the question or chat window of the GoToWebinar interface. We already have quite a few questions, so we won't have time to address all of those today. But any questions that we don't manage to get to, we will answer by email in the next week or so. So a couple of questions that have come in from the audience. First of all, what's the biggest limitation of serial block face SEM technique?
Okay. One, I would say, but which is not a bottleneck for us, it's maybe to achieve lower resolution in Z, meaning cutting slices of lower than 25 nanometer. But I think now more people developing also some ways to overpass this limitation. Another limitation I would see is the size, the width of the sample. It's restricted to the size or the width of the knife. I think it's like 1.5 millimeter. So unfortunately, we can not cut sample that are larger than 1.5 millimeter. Also, one of the disadvantage compared to serial sectioning, TEM or atom SEM, is that you lose the sections. Once they are imaged, they are lost forever. And that's if you want to do additional images or imaging, or if you need them to restain them, it's no way. But I mean, here I don't consider that this as a limitation, because depend on what is on the question, on the biological question. I mean, you have to adapt. You have to use the proper technique for the application. So, yeah, I mean, for me, it's for the moment the main advantages.
Okay, thank you. What's the running time of the typical kind of experiment that you, or maybe there isn't such a thing for you as a typical specimen, but what are the typical, or the range maybe, of acquisition times that you run in your facility?
The typical run, I would say for Connectomics, yes, we need to reach at least, I would say, eight weeks. So the more, the better. But for our application, or for Rainer Friedrich application, I think that's around the time we need to acquire for his scientific questions. But sometimes we have also cells, we have other samples, and then it's more days acquisition. But I would say for Connectomics, yes, you can count on weeks, and that could be sometimes challenging.
Okay, thank you. And then a final. Oh, sorry, go on.
No, and since we have this 5 megahertz, we may reduce a lot, by 4 at least, the acquisition.
Okay, that was going to. That's related to the last question that I was going to ask you, actually, from that came in from the audience. In what ways would you could the acquisition be sped up further?
Speed up further. Yeah, it's always, you have to think in advance what resolution you would like to acquire, and adapt the tile size according to that question. And maybe by reducing the size of the tile, increase the size of the tile, sorry, the opposite, that be a way to speed up the acquisition. So maybe for, let's say, some biological information, you do not require so high resolution, and then you can play with these parameters of imaging. And also, as I said, speed up the acquisition. Maybe now we have the 5 megahertz system, and why not to have a 10 mega or 20 megahertz system? But probably very much challenging to keep the nice single noise ratio or the resolution. That's, yeah, but that could be one.
That's always a challenge.
A challenge, yes.
Okay. Okay. Well, I'd like to thank you again for presenting this webinar today. I'm sure that the audience, I know that I find it very interesting, and I'm sure that the audience did as well, judging by the number of questions that have come in. So thank you.
Thank you, and sorry for overpassing the time.
It's not a problem. Okay. Thank you, everyone.
You're welcome. Bye-bye.